Hello everyone, my name is Miriam Maria, and today I will be presenting a fascinating paper that is titled Gut Microbiome Strain Sharing Between Isolated Village Social Networks. It was first published online on 2024 and then it was published by Nature on 2025. But before jumping in, I want to share why I choose this paper. So Honduras actually has a special place in my heart. It's the reason I got my passport back in 2016, and I have made a lot of close friends there and even coached some basketball teams from the country. So when I saw this study I was conducted in rural Honduran villages, I felt instantly drawn by it. This paper pushed the frontier of microbiome research by showing how our relationships beyond just our households or families can shape the microscopic communities living in our gut. This is particularly relevant in today's world, where we are recognizing that our environments, our communities, and even our social ties can affect our health in unexpected ways. I will walk you through the background, methods, major findings, and overall results. And I want to mention that I am in Puerto Rico right now, so you might hear some cookies, even if I have my windows closed. And I have been here for some time now after the military, so my English is a little rusty. But let's go. For the bottom line up front, here's the key takeaway. Even in rural villages with limited mobility and minimal infrastructure, gut microbiome strains are shared through everyday social interactions like meals, greeting, and free time, not just cohabitation or kinship. This expands how we think about microbial transmission, no longer just confined to households or close relatives. Strain level metagenomics proves social relationships are a microbial highway. This is a paradigm shift. Our microbiome isn't just a product of what we eat or where we live. It reflects who we spend our time with. So, in our class right now, the microbiome, we learn and we know that the gut microbiome is crucial to human health, affecting digestion, affecting immunity, and even mental health. Most past research focus on vertical transmission, meaning from parent to child, household environments, and diets. But this paper asks, what if microbes move through friendship networks? I'm going to repeat that. What if microbes move through friendship networks? It's a bold question, especially in isolated settings like Copan, Honduras, where communities have minimal outside contact. This makes it perfect as a natural lab to test social microbiome sharing without the noise of modern urban life. These two questions lie at the heart of the study. First, can gut microbiome strains be transmitted between people who are not related and don't live together? That question challenges the traditional idea that microbiome transmission mostly happens in families or households. Second question: what role do our social behaviors play in shaping our microbiome composition? This isn't just about who we live with, but who we eat with, touch, greet, or spend time with. Everyday social interactions. By asking these questions, the researchers aim to explore whether social closeness, not just physical proximity, can leave a microbial footprint. Why these questions matter? So, to understand why this research is important, we need to consider the bigger picture. The gut microbiome plays a major role in immune function, chronic illness, and how susceptible we are to infections. So, knowing how microbes spread between people is more than just interesting. It can shape public health strategies. Most studies focus on households or families where people live in close quarters. But what about the rest of our social lives, like friends, co-workers, or people we share meals with? If microbes can spread through casual, non-cohabiting interactions, it means we need to rethink how we manage disease transmission and how we design interventions. This could influence everything from outbreak response to antibiotic stewardship in communities. Something that I really appreciated about this paper, it was their ethical approach. How they involve the community, like from testing and treatment of parasites to donating eyeglasses and even training over 100 locals. This paper showed me what responsible science can look like, especially when working in under-resourced communities. We all know that Honduras have many under-resourced community, and to be able to manage to talk with the community, make them feel safe. The ethical approach it was just amazing, and it demonstrates that their work didn't just extract data; it contributed with the well-being of the communities, and that that was powerful. I will read the paper again, and I really enjoy it most because of the ethic that they have through the whole research. For this study, the researchers worked in 18 isolated villages in Copan, Honduras, which allowed them to capture microbial transmission patterns in tight-knit and relatively close communities. They collected 1,787 stool samples and sequenced them using Illumina NovaSeq 6000, a powerful tool for shotgun metagenomics, which sequences all DNA in the sample. This high-resolution method allowed them to identify microbes at the strain level, not just species. To map out social connections, they conducted name generator surveys, asking villagers who they interacted with daily, who they trusted, and who they spent free time with. Then they used strain for precise strain identification. For statistical analysis, they applied mixed effect models and Permanova, both commonly used in microbiome research to account for community structure and repeated measures. The design was people were sampled again 2 years later, allowing them to examine the stability and persistence of social influence on the gut microbiome. Okay, now let's discuss the first figure. So this figure summarizes how interpersonal relationships and physical contact behaviors influence the microbial strain sharing rate in this enduring cohort study. Let's walk through each panel. If you go to the top left, you're going to see the letter A, and this panel shows that strain sharing is highest among people who are partners, with a 13.9%, followed by those in the same building, mothers and children. As relationships become more distant, like non-kin or people from different villages, strain sharing decreases dramatically to as low as 2%. This indicates that physical proximity and social closeness strongly affect microbiome overlap. Then, if we go down and we go to the panels from B to D, we are going to see that in panel B, people who spend time together daily shares more strains than those who interact less often. Then you are going to see on panel C that meal sharing frequency correlates with strain sharing. Daily or weekly meals led to higher microbiome similarity. Then you go down and you see panel D looks at greeting types. So more intimate greetings like a kiss on the cheek or a hug results in higher strain sharing rates, while gestures like waving shows lower rates. Then if we go to the right side and letter E, you're going to see ranks covariates by their predictive importance, using permutation feature importance, the presence of a relationship, and household sharing are the strongest predictors of microbial similarity, even more than age, sex, or wealth. So the key takeaway is that our social behavior, how we live, how we interact, and even greet one another plays a major role in shaping our gut microbiome. Figure 4, I title Effects of Mutual Relationships of Microbial Strain Sharing. And this figure digs deeper into the role of reciprocity in relationships and its impact on microbial strain sharing rates. We see four relationship types along the axis: closest friend, partner, personal or private, and spend free time. Each one is broken down into reciprocated and non reciprocated connections. The red boxes represent non reciprocated relationships, meaning only one person named the other. The blue boxes represent reciprocated relationships where both individuals acknowledge the connection. What we observe is that reciprocated relationships generally show a higher strain sharing rate, especially in the categories of personal, private, and spending free time. That suggests that mutual closeness and bidirectional interaction intensify opportunities for microbial transmission. Interestingly, for partners, the difference in strain sharing between reciprocated and non reciprocated isn't significant, probably because the closeness is high regardless of whether both parents name each other in the survey. The takeaway here is that the quality and mutual recognition of a relationship, not just its label, matters for how much of our microbiome we share with others. When people interact more openly and frequently with each other, they are more likely to exchange microbial strains. Extended data figure 5. feature importance in predicting microbiome-based relationships. This figure shows us which variables are most important when predicting whether a social relationship exists between individuals and this study, based on strain, sharing, and other covariates. Let's start with panel A, which includes all social and family relationships. The top predictor by far is the strain sharing rate, confirming its central role in inferring social connection. Next come household sharing and average age, followed by sex and age difference. Variables like religion, wealth, education, and indigenous status has have much smaller effects. And finally, we see that gut-specific metrics like Bristol stool scale, diet, water source, and medication usage had negligible predictive value in social ties. Now we go to panel B, which isolates non-kin, different household relationships, and we see a similar story. Strain sharing rate remains the strongest predictor, actually stronger here than in the full model. Age, sex, and religion also remain relevant, but again, microbiome health metrics like medication usage and stool characteristics are not significant in predicting relationships. The takeaway is that while many factors were tested, the presence of shared microbial strains is the most powerful signal for identifying human relationships, especially among those not biologically or residentially connected. Microbiome profiling and analysis. Meta Plan 4 or Meta PHLAN4 was used for species level profiling, and strain level sharing was assessed using custom scripts. They didn't just look at species presence, they look at strain level similarity, and that's key. And to avoid noise, they only calculated sharing rates between people who had at least 10 overlapping genome beans. They also used Bray-Curtis and Jaccard indices to measure beta diversity and dissimilarity. The key result was the microbiomes clustered by village. Even though the villages are small and relatively isolated. This suggests village level ecology matters, but social interactions still explain more variation between each village. The paper uses PCOA and Bray-Curtis to demonstrate this, supported by Permanova. P equal to 0.001. The takeaway: microbiomes are spatially and socially structured. In interpreting the results, it's important to highlight three key takeaways. First, the study shows that social relationships are predictive of gut microbiome similarity. This suggests that our microbiome isn't just shaped by diet or genetics, but also by the people we interact with, especially those we spend physical time with or share environments and behaviors. Second, reciprocated relationships those in which both individuals acknowledge each other had stronger microbiome similarity than unreciprocated ones. This implies that mutual and active engagement plays a role in microbial exchange. Third, the study found distinct microbiome clusters across villages. This indicates that environmental and cultural context, even within a single region, can significantly influence microbial communities. Overall, these findings challenge the idea that the microbiome is an isolated personal trait. Instead, is partially socially embedded, shaped not just by who we are, but by who we are connected and how we interact. In conclusion, this study offers compelling evidence that gut microbiome transmission is shaped by social behavior, not just by genetics, family ties, or living together. That's a big shift in how we think about a microbial ecology in humans. Because of this, public health strategies, especially those targeting microbial health or antibiotic resistance, should start considering social network as part of their frameworks, not just biological or environmental factors. And finally, this study opens the door for future research into how social transmission might occur in other microbiome niches, like skin, oral, or respiratory microbiomes, and across different cultures and lifestyles. There's a whole world of microbial dynamics waiting to be explored beyond the gut. To wrap up, here's some discussion questions we can explore together. These are meant to spark conversation and reflection on the broader impact of the study. How could these findings influence how we manage outbreaks of gut transmitted pathogens? Could identifying key social ties help stop the spread? Second one, should social network data be integrated into public health microbiome projects? What are the benefits and challenges? 3. How might urban versus rural environments shape the patterns of microbiomes sharing? Will we see similar results in more industrialized settings? 4. What ethical concerns come up when collecting detailed data on both people's relationship and their stool samples? How should that be handled? And finally, 5. Could these same methods be used to study other microbiomes like skin or other communities? What might we learn by expanding the scope? This is the references I use together with my paper. And before I finish this presentation, I just want to say thank you all so much for watching my presentation and for bearing with me through all of it. I know it was a lot of information, but this research really fascinated me. And I hope I was able to share some of that excitement with you. If you have any questions, if you had thoughts or something didn't quite make sense or you didn't understood, please don't hesitate to reach out or leave a comment. I will be checking personal and I will be more than happy to respond and clarify anything. Again, I truly appreciate your time and feedback. Thank you.